So we have seen how complex, how stable the structure of DNA is. But from what we have discussed, you can find adaptations of DNA, okay? And you can try to describe the structure of DNA and how it is adapted to perform its functions. If you can do that, then you know that you are good. So now from what we have discussed and from what we know and what we've been knowing, we all know that DNA is a genetic material. It is responsible for storing all the information that makes us. But to do that, you know that constantly, every day, our cells do replicate. Okay? Our cells do replicate always. So now, you need to understand how it is done so that you appreciate nature. Because imagine, you know, you want to form, you have cell A and you want to produce cell A. Two cell A's. By which process? By mitosis. Now the question is, you should be as careful as possible so that everything this side, everything in this cell should be the same in this cell too. Okay? The amount of DNA in this cell should be the exact amount of DNA in this cell too. The structure, everything, so that you don't make any mistakes. Okay? Because once you make a mistake, then you pay for it. So therefore, what does that mean? Then it must be some cool process, some good process, some good work must be done for you to successfully copy your DNA into another copy. So that you have two copies of DNA, of the same DNA in the two cells. You want to form these gametes, the sperm cells and the ova. Remember, there are these cells which carry genetic material, okay, from one offspring to another, from one generation to another generation. Now, if you make a mistake, it means that mistake is going to be transmitted to another generation. If the mistake is harmful, then it means probably the offspring won't be able to survive. So therefore, it must be some good process that occurs with care. And actually, it is. Now for the gamers, this process can make some good video game. Okay? In fact, we should develop a program, okay, where we can create a video game for DNA replication. It is really good. It's really a good video game. So it is a unique, our DNA is some unique molecule that is able to replicate itself exactly without making any mistakes. And by the way, if any mistake occurs, then you must pay for it. It occurs during the interphase of cell division, prior cell cycle. It has that interphase, which is actually the longest stage. It is actually the longest stage. And in this stage, you may assume the cell is just at rest. The cell is just dormant. But when actually the cell is undergoing vital processes. Okay? And one of them is cell division. Now, one of them is DNA replication. So you must be able to copy the DNA so that you produce the two copies of the same, which will be transferred to the next cell. So it is essential for continuity of life. So as we all know, it is a physiological activity, and we've studied that one of functions of globular proteins is they do control all physiological activities within our bodies. So similarly here, the enzymes play an important part. Actually, I said it's a video game. Yes, it is a video game, but the actors, the characters, which other word should I use? They are the enzymes, in fact. If you know what these enzymes do, as far as replication is concerned, then you know DNA replication. Now, there are 4 main types of enzymes that play an important part as far as DNA replication is concerned. And the first one is enzyme DNA helicase. Okay? DNA helicase. Okay? That is the very first enzyme. And this enzyme plays an important part as far as DNA replication is concerned. DNA helicase. Now, what does it do? Now, we have said that our structure of DNA is wound, strongly wound. Wound, it comes from wind. So it is strongly wound. Okay? Strongly wound. Okay? Let me draw this. So it is strongly wound with strong hydrogen bonds between the organic bases inside. But now you want to copy these strands. You want to copy and make the exact DNA, the exact copy. So it means you have to first unwind this DNA to expose these strands so that you copy the DNA. Now the enzyme that is responsible for that, it is DNA helicase. And I think helicase comes from a helix. Okay? It is DNA helicase that is responsible for unwinding the DNA. By breaking these hydrogen bonds, by breaking these hydrogen bonds so that you expose these nucleotides. You expose these organic bases so that you can easily copy the DNA. So that is DNA helicase. Now another beautiful enzyme is DNA primase. Okay? DNA primase. Sorry. Now, what does primase do? Now, from this drawing of mine, from this picture of mine, I think you are able to tell the role of DNA primase. You can see it has deca flag. Okay, it is an enzyme. So if I assume this to be an enzyme, it has some flag. Okay? Now, after you have unwound the DNA, now this DNA, it is some complex stuff that you cannot copy it just in flagranto deligito. Just that's the word we use. You cannot just copy it. Okay, you must have where to start from. Okay, you must have where to start from. Now this DNA primase, as DNA helicase unwinds this double helix, then this DNA primase, it comes with its flag and inserts it where you are going to begin from adding nucleotides or copying the DNA. And this flag is called a primer. Now primers, these are short segments of RNA. Okay? Primers. They are short segments of RNA that initiate the process of adding nucleotides together. So after you've unwound, then this DNA primase, it will come and add this short segment of RNA. Okay? It will add it on the position where you're supposed to start from. Okay? Now, after you've added a primer, then DNA polymerase, that's when it is going to understand that, oh, now this one is the builder. That, oh, so I'm supposed to begin from here, eh? Then that's when it is going to come and add these nucleotides. Okay? Otherwise, this DNA polymerase cannot add nucleotides just in space. It must have where to begin from. It cannot add nucleotides just in space. Okay? Yes. And to confirm that, you can read about, you Google about this word, telomeres. Telomeres. Okay? Yes, you will be happy. That you'll be happy to understand that this polymerase cannot just add nucleotides just in space. So it must have where to start from. And it starts from a primer, which is a short segment of RNA, and it synthesizes a new strand of DNA. Okay? So DNA polymerase is a builder. It is the one that actually does the polymerization. The addition of these nucleotides together, okay, to form a single strand, a complementary strand of DNA. Now, this enzyme DNA polymerase, it adds nucleotide on the 3' end. Now, this is my strand of DNA. Okay, you know my strand, it has the 3' end, and I showed you what the 3' end is and the 5' end. Okay, the 3' end, it is, okay, let me draw my pentose sugar again. Okay, this is oxygen, and this is carbon, and this is CH2OH. Now this is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4, and this one is carbon number 5. Now this is carbon number 3, and this is carbon number 5. So this is carbon 3, and we call it the 3' end. And this is the carbon 5, and we call it the 5' end. Now this enzyme DNA polymerase, it adds nucleotides on the 3' end. Okay? On the 3' end. If it's a primer, it would add the nucleotides on the 3' end of this primer so that you initiate the process. But now see, if this is the 3' end, this will be the 5' end, and we have said that the two strands of DNA, they run in opposite directions. So if this is 5, then this one becomes 5 prime. So if this is 5, this is 3. So the DNA runs in opposite direction. So what does that mean? If this is the 5 prime, then the primer that will be added, this will be the 3 prime, this will be the 5 prime. If this is the 3, 5, the primer that will be added, if you unwind, okay, this will be 3 and this will be 5 prime, 3 prime end. Okay, the same applies. If this is 3, then this one will be 5 prime end, and this one will be the 3 prime end. But if this is 5, then this one will be the 3 prime end. Now you are unwinding, going this side. Okay, you are unwinding towards this side. This is the part that you unwind. Now as you unwind by DNA helicase, then DNA primase is going to add these short segments of RNA molecules, which are primers. Now DNA polymerase, it will add nucleotides there and then. But DNA polymerase adds nucleotides on the 3 prime end. So therefore, if this is the 5 prime end and this is the 3 prime end, it will add nucleotides here. Add here, add here, add here, add here, add here. So as you unwind this part, then DNA polymerase adds on this strand. But look at this strand. If you unwind here, then the DNA primase adds a nucleotide. This is going to be the 5 prime end, and this one will be the 3 prime end. But polymerase adds on the 3 prime end. So therefore you will unwind and polymerase adds in this direction. Then you unwind this part, you add the primer, and then polymerase adds following this direction. When it reaches here, it stops. The same applies, the same applies, the same. So as a result, one strand is going to be continuous, another strand is going to be, there will be fragments, and that these fragments are the ones that we refer to as Okazaki fragments. Okazaki fragments. Okay? And somebody was like, Kaza what? Okay, the students I teach, they know. Those students, they were kind of funny. So these are the Okazaki fragments because they are in parts. So it adds, then jumps, then adds, then jumps, then adds, then jumps. So you form DNA in the form of fragments. But the other one will be continuous. So now that means, but your DNA must also be continuous. So that means there is a need of binding these fragments. Now that's where the binder is going to come from. You see, the gluer, it has the glue. DNA ligase. It is now the one responsible for binding these Okazaki fragments to form a unit strand of DNA. And all this occurs at the same time. You can have a look at this. So I want you to have a look at this. So all these are enzymes that are responsible in copying the DNA. So you can have a look. That is your double strand. So there is an enzyme that is responsible for unwinding. Okay? There is an enzyme responsible for adding primers. There is an enzyme that is responsible for adding nucleotides together to form that strand of DNA. There is also an enzyme that is responsible for gluing. So all these enzymes, they play to form that complex, and the complex is referred to as a replication form. So you can have a look at it. This is our replication form. So we have seen the roles of enzymes, and once you know the role of these enzymes in DNA replication, if you know the roles of these enzymes, then you know what DNA replication is about. You know the entire process. Everything is just a walkover. So it begins with unzipping or unwinding of the double helix, which is done by DNA helicase. Okay? So you unwind the unzipping enzyme. You unwind by breaking the hydrogen bonds that bind these helices together. Okay? And then after unwinding, you'll be exposing these nucleotides. You'll be exposing these organic bases. Okay? So that you easily add nucleotides on these organic bases. So the DNA helicase, it is going to break the hydrogen bonds, okay, that bind these two strands together, so that you expose them. Okay, you expose these strands. Now after exposing them, then the other flag enzyme, the enzyme with a flag, DNA primase, is going to come in, and you add that primer. Okay, the primase tells you that it's over here. Start from here, here. Okay, over here. Start from here. Otherwise, if these primers are not there, then DNA polymerase cannot just add nucleotides in the air. And it has been experimented. If you are in a molecular biology lab, and you don't provide these primers, you cannot carry out the test. You know, they were testing coronavirus with the polymerase chain reaction. Okay, so if you don't provide specific primers, you cannot get anything. You can't see anything. So that one confirms that really you must first provide specific primers for this enzyme DNA polymerase to add nucleotides together. Otherwise, if you don't, then you cannot see anything. Then after adding these primers, then DNA polymerase is going to come and add these nucleotides together. Okay? To add nucleotides. And the adding, like I told you, in one strand, the adding will be continuous. It will be continuous. And that strand where addition of nucleotides is continuous is a leading strand. Whereas that strand where, like I told you, it only adds on the 3' end. Okay? So, in one strand, the addition will be continuous, but in another strand, you will add, and then, now imagine, you've unzipped here, and then you've added the primer. Now you are adding in this direction, because this is where the 3' end will be. So you add following this direction. If you reach here, you stop. It means the polymerase, it will have to jump again and begin from where it has been unwound, where a primer has been added, and you also add again. So as a result, you form fragments of DNA. And those fragments are the ones that we refer to as Okazaki fragments. Now the DNA ligase, the gluer, is the one that will be responsible now to bind these fragments together so that you form that single strand, single continuous strand of DNA. And the whole of this setup is called a replication fork, and it occurs within eukaryotic cells. But for prokaryotic cells, such as bacteria, for them they do form a replication bubble, and very many replication bubbles can be formed. So you can find out the difference between a replication fork and a replication bubble. So by that, then you'll be able to produce the two strands of DNA, where one is a template strand, you can see. So this is a template strand, and this one is a complementary strand. So this one, therefore, is going to be identical to this. Are you seeing? And this, the new strand that is formed here, is going to be identical to this. Why am I saying identical? Because addition of these nucleotides is complementary. Strand A, base A, you add T. Where you have T, you add A. Where you have C, you add there G. And where you have G, you add there C. Addition of these nucleotides is complementary. In that, have a look at this. Assuming this strand here, okay, this strand, let me copy this from here and I put it here. So assuming this strand, maybe it has this sequence, it has A, it has G, it has C, it has T, it has A, it has A, okay, it has T, it has G, it has C, it has G, and finally it has T. So this sequence, and this is my gene that codes for a particular protein. Then as you are adding these nucleotides, you remember you'll be having a pool of nucleotides within this nucleus. You'll be having a pool of nucleotides. So if you want to get a nucleotide with base adenine, then it will be available. If you want to get a nucleotide with base cytosine, it will also be available. You have a pool of nucleotides. Now, as this DNA polymerase, we have pol 1 and pol 3. However, no one is going to ask you this per now. But as you go up, you find them. So as you are adding these nucleotides, they are added in a complementary manner. You follow the base pairing rule, that no mistake is made. If you make any mistake, then that mistake is manifested in the phenotype of an organism. So whereby, where there is base A, then you shall add there, what are we going to add? Maybe, let me change the pen. Let me use, yellow. No, yellow want to be seen in white. Let me use this. So where there is base A, we shall add there, yes, base T. Where there is base G, we shall add there C. Here where we have C, on the complementary strand of DNA, this one, the blue one, I'm now trying to synthesize a new strand. So where there is base C, then we shall add, or our DNA polymerase is going to add base G. Where there is base T, I'm trying to synthesize a new strand of DNA. So here I'll add A. Here there is A, so I'm going to add T. There is A, I'm going to add another T. There is A, I'm going to add A. Sorry, there is T, I'm going to add A. Here we have C, so I'm going to add G. there is C, I'm going to add G. And this one is C, I'll add C. And this one is T, I'm going to add A. So that the new strand of DNA is complementary. And also remember during pairing, when you were pairing these two, when they were pairing, they also paired in a complementary manner. So even when you are adding this, when you are forming this new strand of DNA, it is also formed in a complementary manner. So now, do you agree that this strand is exactly the same as this? And this strand here, this newly formed strand, is also exactly the same as this? Yes. So if you know that, then you are good to go. However, when we are forming RNA, it will be kind of different, and we are going to see how different it is. Not so, guys? Yes. The video is long, but it's worth it. I couldn't split it. It's long, but it's worth it. Now, there are theories which explain what will happen after you've synthesized these new strands. Okay? Now, number one, they can follow the semi-conservative hypothesis. Number two, you can follow the conservative hypothesis or the disruptive, the dispersive hypothesis. So let's see what these ones mean.